All right, so hi, I'm LaBarbera. I'm at the Museum of Vertebrate Zoology, and I'm going to talk to you about how the effects of environment on cell morphology vary among various things. So when we think about selection occurring on a trait, this is maybe the first initial mental diagram you have. There's a trait and there's selection. But if you think about it, of course, the trait doesn't arrive out of nowhere with infinite variation. Of course, selection can only act on variation that exists already, and that existing variation is affected by past selection. So we should really add in the ghosts of past selection because they're also influencing our trait. Then if you think about it more, most phenotypic traits aren't one big, indivisible trait. There's generally different components of that trait that may vary, maybe not completely independently, but not completely correlated either. So even something as simple as, say, limb length, there's lower limb length and upper limb length. There's different pieces of it. So we might split our trait into little mini traits. And then, of course, you want to think about, okay, these different selection pressures may be having different effects on these different traits. And suddenly our diagram has gotten much more complicated. So what I'm interested in doing is looking at if we can take this complicated looking diagram and actually see these relationships in a system, and preferably a fairly simple to understand system, and look at, can we tease apart the separate influences of current and past selection on different aspects of what we might initially think is a single trait. So this is my chosen study system. This very handsome bird is the dark-eyed junco. They are widespread across North America. They're very numerous. They're very successful in a big range of environmental conditions, which makes them well suited to studying the effects of environment on things like morphology. The specific trait I'm looking at is the bill, so it's fantastic that there was Chris's talk and not my talk, because he's already gotten excited about the avian bill. Oh, and so I do this work on collections, so dead birds, but I wanted to show you a live bird because they look much better alive. So this is what they actually look like. So the bill is really important for birds. Of course, it's a tool for foraging. That's probably the first thing you think of. They use it to obtain food and manipulate food. It's also a big source of heat dissipation for birds. It's been shown mostly in the green bird lab. There's no feathers on the bill, and if you look at heat images of birds, the bird is mostly blue and then the bill is glowing bright red because it's losing heat. So this can be good or bad. If you're in a cold place, you might want to keep your heat. If you're in a warm place, you might want to get rid of your heat. But if we're thinking about traits responding to the environment, and we want to think about the two main axes of environmental variation, usually we think about precipitation and temperature. And if we want to match those up to the bill, this works out really nicely. Because we would predict that precipitation will primarily affect the food resources of birds, right? Precipitation is going to affect seeds, it's going to affect arthropods, and so we would expect precipitation to primarily affect the bill through its function as a tool for foraging. Meanwhile, we expect temperature to affect the bill through its function as a source of heat loss. So again, in cold climates, you want to keep your heat, in warm climates, you might want to dissipate heat. So, to measure bills, I did the really basic thing that almost anyone doing avian work does. As Chris said, these are linear measurements of nonlinear things, so there's a little bit of error, but we don't have a 3D scanner. And also it captures a lot of variation, it really does. So we do length, width, and depth, and then from these three measurements, you can also calculate bill surface area. So you end up with 4 features of this trait, essentially: length, width, depth, and surface area. And these are what the birds that I measure actually look like. There's lots of them. I measure them at the Museum of Vertebrate Zoology, which is where I'm based, and also at the California Academy of Sciences. It's really fun to get to hold these birds that are from many decades ago. After a little while, you've been only hanging out with dead birds for a few days. You start to get a little strange. You start to feel like they're kind of making faces at you. You start to take selfies with them. You get a little weird. You need to go hang out with some people. But in the end, I ended up with over 1,000 birds measured. I've split them into summering and wintering birds because the juncos migrate. So if you find a bird in a place in the winter, you shouldn't look at the environment there in the summer because the bird won't be there in the summer, he's going to move. So we split into summer and wintering birds. This is a map of where our points are, color-coded by season. The white ones are summer. I apologize, it's a little hard to see some of the white ones, but the main takeaway is they're well spread out all over California, and also that they're not spatially segregated in terms of the summer and winter division. Most of these birds were collected between 1900 and 1940. That's just when people were collecting a lot of juncos in California. We do have some more recent ones also. And to match these birds with environmental variables that match where they were and when they were, we use the PRISM historical climate data set. So this lets us take a bird collected in 1940 at a specific location and look at what the environment was like in 1940 there and in 1939 there and so on backwards, and get a sense of what that bird was experiencing in its life and also what its ancestral populations might have been experiencing. So the structure of the analysis we did has several moving parts, but we have our 4 bill traits, so length, width, depth, and surface area. We then have 3 environmental variables, mean precipitation, minimum temperature, and maximum temperature. Initially, we had minimum and maximum precipitation, but in California, minimum precipitation is almost always 0, so mean precipitation turns out to be more meaningful. We have 5 different time scales. I just wanted a big, broad range of time scales to try to capture everything we could. So 1 month, 1 year, 5 years, 10 years, 20, or 19 years, not quite 20 years. 1 month and 1 year are too short for evolution to be occurring to change these bills. That's going to be plasticity. The bill is actually not a completely fixed trait. It can change slightly in size and shape, even over the course of a year. So we do expect to see some plastic responses in bill shape to the environment. These birds can breed after they're a year old, so generation time is about a year. So by the time we're getting up to 5, 10, 19 years, we're getting into multiple generations of juncos. And then we have the 2 seasons, summer and winter. And this was all, I don't want to go into all the stats, but talk to me afterward if you're interested. But generalized additive mixed models, we corrected for having lots of p-values, because we had a lot of p-values. Yes. So I'm going to walk through the results step by step, because as you can see, there's a lot of pieces. So to start, with season, we actually found no significant relationships between environment and bill morphology in the winter. This surprised me a little bit, but there you go. In the summer, we found many significant relationships. So if we focus in on the summer, precipitation was not significantly related to any bill trait. This was a surprise. If you remember, we thought precipitation was going to affect the food supply, which was then going to affect the bill. This is what the Grants found in their really famous Darwin's Finches study, right? There was different precipitation regimes led to different seeds of different hardnesses, which then led to bill evolution. So the fact that this isn't happening in our birds is puzzling and interesting, and I have some ideas, but I won't go into now. We found minimum temperature and maximum temperature were both related to bill traits. They both are related to length and depth, but differ in whether they're related to width or surface area. So if we zoom in again, so summer and minimum temperature, we see that minimum temperature is significantly related to bill length at the 1 month scale, that really short plasticity type scale. It's related to depth at all of our longer term scales, 5, 10, and 20 years. And it's related to bill width at the 5-year scale. So notice that there's a lot of variation in scale here. We're not all just seeing the big scales and the little scales. So even though these aspects of the bill are related to technically the same variable, right, minimum temperature, they're actually related to quite different things because they're at different scales. You get the same thing when you look at our results for summer and maximum temperature. So bill length is related to maximum temperature at the 1 month scale. Bill depth is related to it at the 19-year scale. And especially notice bill surface area is related to Tmax at the 1 month scale. And you might predict that bill surface area, because that would be related to something like heat loss, because you lose heat through your surface area, that it would be positively related to maximum temperature. You want to lose more heat as you get hotter. And in fact, that is what we see. This is a graph of maximum temperature on the x-axis and surface area on the y-axis, and you can see it goes up in the right direction, which is reassuring. The conclusions we can draw, it matters what season you're looking at. I will say I can't tease apart whether this is season or life stage, because these birds only breed in the summer. So if you're looking at a summer bird, you're also looking at a breeding bird. If you're looking at a wintering bird, you're also looking at a non-breeding bird. With this data set, I can't tease those apart, but season and or life stage matter. Environmental variable matters. Probably no one is surprised to learn that precipitation and temperature are not the same thing, but there you go, they're not the same thing. The specific aspect that you're looking at of bill morphology matters. So even given something that you might think is a single trait, these different components of it do respond to different pressures. And then finally, temporal scale does matter. Again, we see different relationships at different temporal scales. So if we want to make that a little broader, you can just say that different selection pressures may act on different aspects of a given trait at different times of the year and over different temporal scales. So thank you.